Yes.
All right, perfect. Okay, so hi guys, thanks for coming. I'm Graham. I'm going to be talking about these two papers that came out of the Way Lab in Michigan State. I think the papers are there's a lot of information in them, so I'm not going to talk about everything. Mostly, I'm going to be focusing on their representations of protein structure. So I'm not really going to get too far into the machine learning. I know they use a lot of techniques. So I'm happy to discuss that after, but that's not going to be my main focus. In general, I'd like this to be, a discussion. So please, if I'm confusing or if I make a mistake, don't hesitate to chime in. That'll be helpful. Okay, so at a high level, I think these papers are interesting because of the representation of proteins that they're using. So I'd say the overarching point goes something like typical geometric representations of proteins tend to be often difficult to compute and kind of complex. And so maybe those aren't ideal for machine learning tasks. And so the question that I think they're trying to answer is, can we develop a representation of proteins that retains this information content? Yeah.
Graham, I'm sorry, there's a question from the class. If you look at your screen, you'll see that you have little videos of, you and me and Andy.
Yeah, I was wondering about that.
I think if you look at the buttons, I think you can minimize those.
Yeah.
All right.
I don't know if I can get it away completely, but
I think minimizing is good, and you can move it up there. Okay, so is that better?
Yeah, I'm going to put it in the bottom left because I used the top.
Yeah. Okay, so so Faraz and Ricky said that's perfect. Great. So good use of the technology here, everybody. That's good use of chat. So chat worked really well, by the way. So what I can tell is that with the raising your hand, I can only see that if I tap participants, which is actually useful. But the chats actually come through as little bubbles for me. I don't know if that works for everybody. So that definitely feel free to break through that way. So, okay. Thank you all for helping with that. Kudos. And please continue, Graham. Sorry for the interruption.
No, thank you. Right. So the question is, can we get a good representation of proteins that also reduces the complexity here? And so the idea that's developed through these papers is this idea of using topology and specifically persistent homology to get representations of proteins that avoid some of this complexity. And the question is, are these good representations for learning? Okay, so essentially what I'm going to talk about through this talk is I'm going to start off by giving you some background on algebraic topology and persistent homology. And then I'm going to talk about the papers individually. And I'll note that this view of algebraic topology is going to be very simplified. It can be a very abstract topic, although it can be quite easy to implement. I think Bruce put some really great resources on the Piazza. But I guess the point is that what I'm giving you today on algebraic topology is really only going to be scratching the surface, but hopefully it's useful for understanding the paper. Okay, and I'll also mention that if you're interested in learning more about computational topology, in addition to the ASMB chapter 50, which is excellent, Herbert Edelsbrenner has a really great book with a really great cover that I found really useful when putting this together and learning about this topic. So highly recommend. Okay, so we're going to start with some definitions that'll be really critical for our understanding of what's going on with algebraic topology here. The first thing is that we're introducing this idea of a K-simplex. So we're defining that as the convex hull of a set of affinely independent points. For those of you who don't want to deal with affine geometry, this is essentially equivalent if you pick one of the points and convert all of the rest of the points to vectors, it's linear independence. But here I'm showing you some examples of simplexes or simplices. A zero simplex would just be a vertex. This one simplex is a line, two simplex is a triangle, and the three simplex would be a tetrahedron. And I'll note that the interior of the tetrahedron is also included in this definition of a simplex. And of course, you can go up to higher dimensions, but we're going to be staying in these low dimensions for now and also for the papers. Okay, and so when we're working with these simplexes or simplices, it's useful to define some idea of a face. So formally, it's defined as the convex hull of any proper subset. And so, for instance, a zero face of a line would be a vertex. One face of a triangle is a line. Two face of a tetrahedron is a triangle. And I'll note, just for those of you who are interested, generally you attach coefficients to each simplex and face. But for the purposes of this talk and also for the papers, we're going to be working with those coefficients being 0 or 1. And so I'm going to be ignoring the coefficients mostly, and we're just going to think of it as existence or not existence. Okay, and so moving on, this concept of a boundary is quite useful. For a simplex, you can just define it as the union of all faces. So the boundary for the 0 simplex or the vertex is just null. For 1 simplex, it's the vertex endpoints. For a 2 simplex, it would be the triangle, and for this 3 simplex, it'd be the exterior of the tetrahedron. And so this is obviously there's formal ways to define it, but I think the concept of a boundary is pretty intuitive in these cases, at least. And then complementary to the boundary, the interior of the simplex is just everything except for the boundary. So here I'm showing you the interiors of these simplices, also fairly intuitive. And so this brings us to this idea of a simplicial complex. And I'm oversimplifying here, but for our purposes, we'll think of it as just a collection of simplices with the caveat that they need to intersect nicely. And you can think about the dimension of a simplicial complex as the maximum dimension of any of the simplices. Here I'm showing you a 3 complex. And so these are the objects that we're going to be working on mostly throughout this talk and also in the paper. So some more terminology related to simplicial complexes. There's this idea of a chain. So a P-chain is a formal sum, and because we're working with coefficients of 0 and 1, we can just think of it as a set of P simplices in K. So we're going to be using this example simplex. Can you guys see my mouse, actually? That would be useful. Can you guys see my
Graham,
mouse?
I think you have to use your the laser pointer in presenter mode.
Okay, I don't actually have presenter mode. That's okay. I don't actually need the mouse. So on the left, we have this example simplicial complex, which we'll call K. This is going to be a 2 complex because the biggest simplex in that complex is of dimension 2. And then in the center, we have one of the 2 chains of K, and that's simply excluding, it's made up of only the 2 simplices in K. And then on the far right, we have yet another 2 chain of K. So there are multiple different 2 chains of K. The point is that it only contains 2 simplexes, 2 being the dimension of the simplex. And another thing that's useful to define is this idea of addition of different P chains. So for instance, for our purposes, we can think of addition as the symmetric difference operator. And so the idea then is if you add the 2 simplexes, the two chains on the right, you would get this triangle that is the difference between them. This 2 simplex that is the difference between them. And so a critical point here is that the P chains with, in our case, the symmetric difference operator form a group. And so that means if you add two chains, you will get another 2 chain as a result. So before we had this idea of a boundary of a simplex, and so now we have to start thinking about what the boundary of a chain would be. And so we define this idea of the boundary operator. Formally, in order to take the boundary of a P chain, you take the formal sum of all the P-1 chains. And so this is why this line disappears when you take the boundary operator. But essentially, it's a very intuitive idea. You just take the boundary, and so you can apply this operator to map the P chains to the P-1 chains. questions about this? This is a pretty critical point to understand.
Graham, I'll just ask, well, maybe a general question. Does everyone know what a formal sum means?
My understanding of a formal sum is that it's a generalization of addition. But I think my understanding of the technical distinctions might be a little unclear, or at least not clear enough to explain it well. Would you want to explain it?
I mean, it's pretty much it's just a generalization of addition, but it's like, you know, if you want to add two things that are not numbers, how do you do that? So like, if I have, you know, triangle plus a square, what is a triangle plus a square? I could say just formally, right? I could say triangle plus square is this thing, which is a triangle plus a square. So it's kind of like tautological, but that's
right.
Basically just like define, I know I can sum things like triangles, right? I can sum up triangles and get something, which is, you know, a chain or a complex.
Yeah. 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 And again, in our case, so we're working with addition modulo 2. So essentially the idea is if you add something to itself, it just disappears, right? Actually, the next slide might make this a little more clear as well. Because we'll talk about the idea of a cycle. So a P cycle is a P.
Graham, I think another way,
Yeah.
I think really like this discussion. I think another way is that, you know, adding, like Ricky said, adding a triangle and a square together, that's like, you know, triangle plus square. But one way to think about it is, suppose someone gave you one more triangle and two more squares, then your formal sum would be 2 triangles plus 3 squares. So the idea is you can get more of them. And then if someone came and took away one of the squares, then you'd have 2 triangles plus 2 squares. So you can kind of do addition on these things in some sense. And I think that was in there. And there's a question from Faraz. Yes, they're algebraic properties these have to satisfy. So Faraz asked about closure, which is there, but actually you satisfy, it's I guess it's formally an ideal. So you don't have division, but you have plus and minus, and you have an identity and things like that. So there's certain operators that has to have there. I think it's a module over the integers if you're a mathematician. But so that allows you to do operations like that. I think your next slide will make it clear. And I really appreciate you all asking this question and also for answering it. So great job.
Yeah,
I think actually even another way that maybe everyone would be could relate to, a vector space is basically defined this way, right? So like, how do you define a vector? You define it in terms of adding up the components. So first you learn how to add 2 numbers, right? And you define how to add 2 vectors based on the same principle. So if I say, you know, I take the x-axis is an object and the y-axis is an object and the z-axis is an object, then a vector is just kind of a formal sum of these vectors.
Right. Yeah, good points all. Okay, so we want to have this idea of, so there are some chains that behave in special ways. So a P cycle we're defining as a P chain whose boundary is 0. And so here I'm showing you 2 examples of cycles. And the idea is essentially, in order to take the boundary, you sum up all of the P-1 chains, right? So in this case, the P-1 chains would all be vertices. And when you sum them up, you'll notice that each vertex appears an even number of times. So either 2 or 4 times, I think, in these cases. And so because we're working in modulo 2, we're going to, all of these vertexes are going to cancel out and the boundary will be empty. And so this is the characteristic by which we define these cycles. It's useful to note also that the cycles also form a group. So the P chains form a group, the cycles form a subgroup of the P chains, and the boundaries, which are which are essentially, as we've defined them here, right? The boundaries of higher chains form a subgroup of the cycles. And so there's this interesting group structure that I'm showing here, where you can get from the p+1 chains to the p chains and the p chains to the p-1 chains using the boundary operator. And there's this interesting group structure here where you have subgroups of the chains nested in this interesting way. And so I'm going to be denoting the chains with C, the cycles with Z, and the boundaries with B. And so one question that you might ask, and I think it's one of the important ones for talking about topology, is are there cycles that aren't boundaries? And if there are, how many are there? And this is one way to characterize the connectivity of a shape or space. And so to answer this, we introduced this idea of homology groups. And the homology group technically is defined as the pth homology group is defined as the cycle group modulo the boundary group. And the term Betti number refers to the rank of the homology group. I think this is kind of unintuitive as stated, so I'm just going to do a simple example, and I think that'll make this clear. So what we're going to be doing is we're going to try to calculate what the first homology group is for this simplicial complex here. One way that the Betti numbers are commonly explained is that the 0 Betti number counts the number of connected components. The first Betti number counts the number of holes. The second counts the number of voids. And that's, I think, a useful way to think about it for this purpose, although it doesn't always generalize well. But so intuitively, this simplicial complex should have a first Betti number of 1. And so we'll go through how we would do that. And just to mention, if you're getting confused, that simplicial complex is always going to be up in the right-hand corner there, so you can refer to it. Okay, so if we're going to talk about the first cycle group, one useful question to ask is what's the basis for this cycle group? So in the bottom left, I'm showing you the largest possible one chain for the simplicial complex, and on the right, I'm showing you the basis for this cycle group. And the idea is basically that if you combine those 5 elements, any of the 5 elements on the right, you get a cycle, a one cycle in K. And these 5 elements are independent, so you need all of them in order to get any cycle in K. And so we would say that this is a basis for this group. And then similarly, you want to ask what's the basis for the boundary group, for the one boundaries. And so again, on the bottom left, I'm showing you the largest possible 2 chain. And on the right, I'm showing you the basis for all possible boundaries, all possible 1 boundaries in K, right? So again, you need all 4 of those boundaries in order to be able to construct any larger boundary. So this is a basis. And when you ask the question, okay, so what about the first homology group? And what we said before is you're taking the cycles modulo the boundaries. And the idea is here I'm showing you all the cycles, and I've clustered all of the boundaries over to the right. So this sort of intuitively gives us the idea that the basis for the first homology group is this one cycle that isn't a boundary plus all of the boundaries. And so we can call this a homology generator. I think we can also call it a homology class. But this is the only coset in this group. So the first Betti number for the simplicial complex would be 1. And yeah, okay, any questions on that? Essentially, you're looking at, the question you're answering is, how many cycles do we have that aren't boundaries? And it turns out these things are actually pretty easy to compute. And I'll refer you to that Piazza post that Bruce made for that.
That was very intuitive, Graham. Thank you. Are there any questions on this part of the presentation?
Cool.
Nicely done. Thank you.
Thank you. Thank you. Okay, so we've we've gone through all of algebraic topology, so we're experts in that now, and but it isn't quite clear how we would apply this to proteins, right? Because if you think about proteins as they're usually represented, you know, they're fairly simple. They're just atoms connected by bonds. Those aren't going to be very topologically interesting. So we have to figure out some way to generate interesting complexes from our point clouds that we use, or that we think of proteins as. And so one concept that's going to be really useful, again, these things I'm not going to try to define super formally. I'm just going to give you a sense of what they are. The Voronoi diagram is, or a Voronoi cell for a point is the set of all points that are closest to that point. So it's an interesting way to partition a space. And so here I'm showing you an example of a Voronoi diagram in 2D. And you'll have seen this term alpha complex in the papers. And essentially what you do is, for the set of points, you generate a union of balls with the center centered on each point, and you pick a specific radius in order to do this, and you intersect that with the Voronoi diagram. And anytime you have an intersection, you fill in that complex. So an intersection of 2 balls would be a 2 simplex, intersection of 3 balls would be a 3 simplex, and if you have 4, it would be a sorry, I'm I+1 here. So intersection of 2 balls is a 1 simplex, intersection of 3 balls is a 2 simplex, and so on. Okay, and so the critical point here is that if you vary the radius of these balls that you've used to create the alpha complex, this will change the resulting simplicial complex, and therefore this will change what the homology generators are. So instead of making my own demo for this, I've found a great one on the internet. And so here we have a collection of points, and I'm going to be varying this distance parameter alpha. So as we increase alpha, you'll notice the appearance of simplexes in this diagram. Let me know if there are any issues with seeing these, but as I'm increasing the points, you note the appearance of 1 simplexes, increasing the radius, suddenly some 2 simplexes have arisen. And as you keep going, you note the appearance of 2-dimensional holes. And if we were doing this in 3 dimensions, you'd notice voids as well. And so this is what's referred to as the filtration parameter in these papers. And so in doing this, you generate what's called a filtration of alpha complexes. And these are the objects that the paper uses in order to generate all the barcodes. Okay, so that was obviously a simplified view of what they're doing, but let's talk about the persistence barcodes. Okay, so this is from a 2014 paper from the same lab on these barcode representations specifically. On the left, what I'm showing you is, or what they are showing you, is an icosahedron in 3 space, and the radius of these balls represents the alpha parameter. And so focusing just on the left, you notice as you increase the radius of the balls, the components connect, and then at point C, you can't quite see it, but there will be a void in the center of this ball, or of this sphere. And on the right, we're showing the Betty barcodes for this shape. So on the y-axis, they never label the y-axis, but the y-axis, it isn't really a scale. It's just as you go up the y-axis, you have your homology generators. So on top, we have the Betty 0 bars, or the 0th dimensional homology generators. On B1, we have the first Betty number, or the 1-dimensional homology generators. And on B2, we have Betty 2, or the 2-dimensional homology generators. So each horizontal bar represents a different homology generator. And so for Betty 0, you'll notice there are 12 bars. And I should mention the x-axis is going to be the value of your filtration parameter. It's usually in angstroms in this paper. So you'll notice that in this top graph, you've got 12 bars until you get to about 2 angstroms. And this is because until you increase your filtration parameter, the radius of those balls, to about 2 angstroms, none of your vertices connect, right? So you have, until then, you have 12 components. And then at 2 angstroms, the radiuses, the radii of the balls meet, and then from then on, you have 1 connected component. And so that's how you read that top graph. The middle graph, you'll notice there are no bars. That's because there are never any 2-dimensional holes that form. And then on the bottom graph, you'll notice at about 2 angstroms, a void forms. And then as you increase the balls further, it becomes fully connected and that void disappears. So this is the simplest example of the Betty barcodes. Are there any questions on that? I've got some more complex examples that we'll get to in a few slides. But any questions on how to read these diagrams? All right. Okay, so here's maybe a more interesting one. So this barcode diagram was generated using just the C alpha atoms for an alpha helix. And the Betty 0 graph shows similar things until you reach a radius that's about the length of a peptide. All your components are disconnected. And then after that, you have 1 connected component. The Betty 1 bars indicate that at a certain point, you form circles between the 4 consecutive C alpha atoms for a while, and then they disappear. And then the bottom one indicates that you never form any voids. So this is quite simplified. If you look at an alpha helix with all of the atoms included, including the side chains, things get much more complicated. I'm not going to go into that too much. And so this is this idea of a topological fingerprint. The assertion that they're making, and I'm actually not sure how justified it is, but the assertion that they're making is that this provides a fairly unique representation of a protein structure. And it's one that is based on this persistent homology, which has this geometric information embedded in it. And then this is an example from the first paper. So here, what we're showing on the left is the wild type protein, and on the right, we're showing a mutant protein. So you can see the mutated amino acids represented in ball representation. And their point here is that there's a difference between the topological fingerprints generated for these proteins. And in particular, they generated these fingerprints only looking at interactions between the mutated amino acid and the rest of the protein. And one of the things that they highlighted for this figure in particular was that if you focus on the Betty 1 barcode, you notice that the mutant has fewer bars than the wild type. So the mutant's on the right, wild type on the left. And they suggested that this indicates that the mutant doesn't pack as well because it creates fewer ring and cavity contacts. So an example of how these Betty barcodes might differ and also why they might differ. Okay, so all that stuff that I just went over is essentially the background for the papers. And unless anyone has any questions, I'm going to go forward and talk about some of the specific things that they do with it in the 2017 and 2018 papers.
I think that was great, Graham. There was a question from Nicole, if you had just a minute.
Yeah, absolutely.
Yeah, it was just a couple slides back. It was just a clarification. When you said, yeah, that one, when you said you start to see connections when you get to the length of a peptide, did you mean length of a peptide bond?
Not the length of a peptide bond. I meant the length of a, like if you were to measure the length of an entire peptide, I think it's between like 3.6 and 4 angstroms, roughly. I think there's been different estimates of how you look at that because it's not super well defined, but it's basically if you took just one amino acid and measured the ends, you get roughly that length, I think.
Okay. Thank you.
Yep.
Thanks.
Another, I guess a more precise way to say it would be it's about the length between 2 C alpha atoms. in a peptide.
Okay.
Yeah. Cool. Other questions? Cool. Okay. So in the 2017 paper, their overall method, once you understand the ideas behind the persistent homology and this particular representation of proteins, is actually pretty simple. What they do is they select which atoms they would like to include in their point cloud. They define a distance metric. In a lot of cases, this is just your standard Euclidean distance. They use these standard persistent homology ideas to compute the homology generators for different filtration parameters, use this to generate the barcodes, and then they featureize these barcodes and use them, they feed them into their ML techniques, which honestly I'm not going to talk about, and then they use this for prediction. And that's also more or less the method for the 2018 paper, but I thought I'd put it here. Okay, so the first part is the atom selection. Apologies for all the text on this slide, but I couldn't figure out a better way to do it. For the Betty 1 and Betty 2 barcodes, they select carbon atoms only, and they also select all heavy atoms. So they generate barcodes for these things. One of the ideas behind the carbon atoms only was to try to get a sense of nonpolar and van der Waals interactions. And then for the Betty 0 barcodes, they select lots of element pairs. And so as far as I can tell in this paper, they manually select and or take all pairs for protein ligand atoms and then also protein mutant atoms. And so what I mean by that is they only consider interactions typically between a protein and a ligand if they're trying to learn protein ligand binding affinities, and only between the mutation and the rest of the protein if they're trying to predict the change in stability upon mutation. And I think in general they pick all pairs, and so they'll only look at carbon-carbon bonds or carbon-carbon interactions, and so on and so forth. One thing that I'll mention, and this bothered me, they mention in the paper that they don't actually use the barcodes for the Betty 0 features, and instead they only use distances. The exact way that they did that isn't super clear. So that was one gripe I had with the paper, is how they actually computed the Betty 0 barcodes wasn't very clear to me, but that's a minor point. Okay, so the second step is how they define the distance. For the Betty 1 and 2 barcodes, they just use straight Euclidean distance. And then for the Betty 0 interactions, and this was how they ensured that they only looked at protein ligand or mutant rest of the protein pairs, is they just set the distance to infinite unless the atom pair was a protein ligand pair or mutation rest of the protein pair. And one of the reasons that I mention this is that they'll explore this, they'll explore different distance metrics in the 2018 paper. And then, so how do they actually featureize these Betty barcodes? From what I can tell, what they do is they use what's called a counts in bins method. So what they do is they split up their filtration interval into a bunch of smaller intervals, and then they count three things mostly. They count how many bars are born in that interval, how many bars die in that interval, and how many bars are persisting through that interval. And so in this case, in this particular interval here, I've shown that there are 9 bars being born, and as they step their interval across, this interval has 7 bars being born, 3 persisting through it, and 6 dying. And then this last interval is mostly just death. And then they take these and they turn them into feature vectors. So they have a vector of all the born information, death information, and persistence information, and they're using these as their features that they're inputting into machine learning, as far as I can tell. All right. And the results essentially are that they did better than everyone else. So here they tested their prediction of protein ligand binding affinity based on this PDB bind data set. And they find that they do significantly better than most other scores and have a lower RMSE. Then they also test their ability to predict the change in stability of a protein upon mutation. And I will note that although they do better than most people, this TNet MP2 also includes other features like solvent excluded surface area, van der Waals, interactions between atoms, charge, electrostatics, and things like that. So from the point of view of just pure topological representation, they're still close to the top, but they don't do as well as their top performing model. And then one other interesting thing that they did is they used, I believe they called it multitask learning. And so essentially they incorporated membrane proteins are particularly difficult to model because of the different chemical environment. And so interestingly, when they trained their models on both membrane and non-membrane protein stabilities, they did better than the membrane protein data only. So that was pretty interesting. And I think the main observation from this paper, aside from the fact that they're able to get pretty good prediction accuracy using topological features, was that both the Betty 0 and the Betty 1-2 features contributed to the prediction accuracy. Okay, and then the 2018 paper, there's quite a lot there, but I think with respect to the representation of the proteins, I think that there are four main innovations that they presented. The first they call multi-component persistent homology. The second they call multi-level persistent homology. Third is electrostatic persistence. And then they also pursue some different featurization methods that seem to work better. So I'll address these individually. I thought the naming was a little confusing, but that's kind of a minor complaint. Oh, and I should mention, I forgot to mention this. So they use the term element-specific persistent homology a lot. All that refers to is that they're selecting specific, like different element combinations in order to create these barcodes. So that's the whole idea. And the multi-component persistent homology is quite similar. They're still selecting different element combinations. But what they've done is they've selected all of the element combinations, used a separate machine learning model to assess which element combinations are most predictive for different tasks, and they've ordered these element combinations by their importance. Otherwise, the other axis is just the same as those vectors. It's just the filtration parameter. And similarly, they keep track of the persistence, death, and birth for the Betty 0, Betty 1, and Betty 2 numbers. And so these are their 2D image-like representations. Yeah, that additional access is just more element combinations. And they include 16 channels. Those refer to the birth, death, or persistence for the various Betty numbers, as well as features for both the protein ligand complex and the difference between the protein ligand complex and the protein. So these are their 2D representations, and they feed these into a convolutional neural network. Their idea for a multi-level persistent homology is essentially just a modified distance matrix. This is very similar to what they did in order to focus on protein ligand interactions or mutation rest of the protein interactions. What they do here is they set the distance to be infinite if the two atoms are bonded, and it's just Euclidean distance otherwise. The idea here is essentially to de-emphasize the covalent interactions. And the rationale for this is that they show that it can be more sensitive to small changes. So here, this is a figure from the paper. They're showing two slightly different conformations of, I believe that's an alanine, although they're coloring I think they're coloring the hydrogens green. And what they're showing here is in the circles represent the top conformation and the stars represent the bottom conformation. And they've computed these birth and death values based on their multi-level distance matrix. So setting the covalent interactions to be infinity. And they're showing a difference between, essentially what they're showing is that the stars and the circles do not overlay perfectly. And what they said was if you just use a regular Euclidean distance matrix, then the stars and the circles do overlay perfectly. So the idea, I believe, was that this was designed to be more sensitive to small perturbations. And it does indeed seem like this improves prediction accuracy for the higher Betty number features. The third idea is almost exactly the same idea, but instead of changing the distance functions to focus away from the covalent interactions, you change the distance function to essentially be some measure of electrostatics. And here they're using a function. I believe the reason they use this particular functional form is to scale the values to be between 0 and 1. I'm not positive why they're not using, like usually when you see electrostatics, they decrease with the square of the distance, and here they only decrease with the first exponential. So not sure exactly why they're doing that, but the idea is that they're embedding this electrostatics information into the topological fingerprint. And then as far as their additional featurization method, instead of doing this counts and bins method that I showed you before, for some of the barcodes, they instead keep track of the average, standard deviation, max, min, sum, and count of the birth values, depth values, and persistence values for these bars. And again, this seems to yield better performance in some cases. So if we look at the results, they tested some additional things. So one thing that they tested was very similar to the 2017 paper. They tested their prediction of protein ligand binding affinity on multiple different data sets. They did predict on, I believe, the same data set as the 2017 paper, and they made only a very slight improvement there, which I thought was interesting. So this kind of made me question to what extent the improvements in this paper, to what extent they're actually improving their prediction accuracy in this particular task. But again, they beat the other methods they tried. And then I think one of the most interesting parts of this paper was the fact that they adapted their method to do virtual screening for protein ligand binding. So they tested on the directory of usable decoys, which basically has a bunch of proteins and also small molecule ligands that both do and do not bind to the proteins. And then the task is to discriminate between the ligands that bind and the ligands that do not bind. And their best method outperformed all of the other independent methods, and which was pretty cool. Okay, and as far as observations on what worked and what didn't work with respect to their changes and the new features that they introduced, they found that the element combinations for their multi-component persistent homology for more than 2 elements were redundant for the Betty 0 features, but at the very least didn't degrade performance for the higher Betty features. And so this was essentially their rationale for including them. They thought that it might increase the generalizability of their method. So that was one observation they made. And then 2 other things that they noted is that the electrostatics, embedding the electrostatics into these Betty barcodes contributed significantly to accuracy, and that the multi-level distance did very well in computing the Betty 1 and Betty 2 features, not so much for Betty 0, somewhat obviously. And so I think overall, I thought this was an excellent paper. I thought that the topological representation of proteins was really interesting and clearly works well with machine learning. I had a few questions that hopefully you guys can help me answer. I was confused about the fact that they included features for the protein ligand complex, the protein, and also the difference between those two. I would have thought that the difference between those two features would have been just baked into the machine learning model. So I was confused about that. I didn't like that there was some ambiguity in how they constructed the Betty 0 features. As I mentioned in the 2017 paper, they didn't actually use the barcodes for the features. They just used distances, and I wasn't sure whether they did the same thing in the 2018 paper or whether they were actually computing the barcodes. Also, in general, for the 2018 paper, this is both a criticism and something that I praise them for, I think, because they tried a lot of things and they reported, I think, everything they tried, or at least a lot of what they tried. So there's a lot of information in this paper, but at the same time, it makes it difficult to evaluate the contribution of these specific techniques. For instance, their final models that they report essentially every time was their final model was a combination of a bunch of different models. They had a lot of votes in their final model. So that made it difficult to see which techniques were contributing a lot and which techniques might not be contributing very much. And then the one concern I had was that the 2018 paper, they didn't improve that much on the protein ligand binding affinity task on the data set that they tested in the 2017 paper. But I think overall, my criticisms are fairly minor, and I was pretty impressed with their ability to generate this useful representation of proteins with this interesting persistent homology approach. And I think that's all I got. So I'll open it up to questions if you guys want to talk about it. Yeah, I'm interested to hear what you think.
That sounds great, Graham. Can you hear me?
Mm-hmm.
So that was great. I think that really good presentation. Let's have some discussion. I think chat is working quite well. If you have a question, why don't you send a chat message and we'll recognize you, and then you can chat.
Sure. I'm going to stop my screen share just so I can see the chat and things like that. I can put it back up if people want to see slides.
Great. So who has a question or discussion for Graham?
We can talk about the machine learning stuff if people are interested in it as well. I didn't go into it mostly because I'm not very familiar with it, and I was more interested in the persistent homology stuff. But I know that we have some people that are really good machine learning people. So so maybe they can talk more about like the gradient boosting trees and things like that if people have questions.
So a question, something I brought up on Piazza about the complexity. Was there any kind of information about so they did comparisons on accuracy. Was there any comparison on complexity or running time or how long? Because I know I've done a lot of topological data analysis in the past, and often, I guess in this case, you're limiting to only 2 dimensions, so that might not be such a big problem. But if you go to higher dimensions, often the scaling like becomes quite computationally expensive, or at least in practice it runs a while.
Yeah. So they didn't talk at all that I saw about the running time or the time complexity for feature generation. They did mention in the 2018 paper that their 2D image convolutional neural network was too slow to run on the virtual screening task. So those 2D image representations of proteins, I don't know whether that was because it was expensive to generate those features, or whether it was just they were large features and it took a lot of time to learn those parameters. Yeah, I'm not sure. They didn't go into that very much that I saw, and certainly no discussion of the complexity for computing the features. Yeah, I agree. I think it would have been a useful thing to have the runtime of all of those different methods, like in those tables where they compare the accuracies. Because I agree, like if your method takes orders of magnitude more time to get a slight improvement in accuracy, it's questionable whether that's worth it.
Yeah, like I was thinking, if I don't know what exact features they put into the previous version that they mentioned, but when they're saying they're doing these like quantum mechanical calculations or other things, like I don't know if that's very local, or like I could imagine, right, like doing a lot of calculations on energies that are just like n squared maybe at most, right? And then right. I think certainly you'd have at least one, maybe more factors of n that show up in computing the topology stuff.
Yeah.
Ricky,
yeah, I don't know.
Ricky, what do you lose by going to the Z-mod 2 homology as opposed to integral homology? What's the
it depends on the topological space you're looking at. Sometimes you don't lose anything. Sometimes you do.
Is integral homology the most sensitive in general compared to the over rationals or something else, or Z-mod 2?
Not always. Let me think for a moment. I mean, so if you're just comparing to, I think, yeah, like generally, if you compare like a finite ring to a larger ring, oftentimes you'll go up in the kind of information that you get, but not always. Like if you go all the way to R, right, then you end up with a slightly different situation than if you're looking at Z. Like if you look at real number coefficients, you get different things than integral coefficients. In this case, I don't think you would really get much benefit from using different coefficients, but I'd have to think about that a little bit more to actually be sure of that.
Weird things that aren't embeddable, like with torsion groups and stuff, those would distinguish between more spaces, right? If you had, non-orientable things and Klein bottles and stuff like that.
Right. But since we're looking at like just a point cloud in 3 dimensions, I don't think there's really much that you lose between different choices.
Yeah.
I have a question. So Graham, you said that they used the difference between were you confused as to like why they used the difference between the protein, the complex, and the difference, or just the complex and the difference?
All three.
Yeah, I think they
use all three. So if you look at, I'm looking at which paper is this? I'm looking at the 2017 paper.
Yeah, Oh,
maybe they didn't do this in the 2018 paper.
Oh, okay. So the 2018 paper, they don't do it. That was
Okay.
why I got confused. Yeah. In the 2018 paper, they don't do it.
Okay. Okay, so maybe that's just something that they just had in there just because in the 2017 paper and thought about it and took it out. Because definitely in the 2017 paper, they have the interactions for the protein ligand complex, the protein, and also the difference between the protein and the protein ligand complex.
Okay.
So, yeah.
So yeah, I bet they just had redundant. It was a redundant feature that they got rid of later.
Yeah.
What it looks like. So your suspicion is correct that there's no reason to do something like that.
Okay, cool. Yeah, I was confused. That seems like the sort of thing that should be taken care of.
Yeah. So,
I have a question that maybe I could interject if we could do it. So it has to do with the featurization, which I have to admit I understand better for the 2017 paper than for the other ones.
Yeah.
But so one of the things is that these things, as I understand, are basically counts of birth, death, and persistences, the VD, P, and so forth. So I'm just trying to get an idea, I mean, of y'all's intuition about this. So I'm always interested in these cases of what two things could you confuse. In other words, so the thing, I mean, for those of you who haven't thought of this before, the idea is that different spaces, in general, you'd like them to have different topological invariants, and none of them are perfect. But generally, we can have the case where two spaces that are different will have different topological invariants, but you might not discriminate enough. So you might have two spaces that are a little bit different, but have the same topological invariants because it's not a perfect classification. So in this case, because you're just looking at the counts, you know, in some sense, you could have a thing that at a certain point in the vector had the birth of 3 homology generators. And that could therefore look like it's similar in the vectorization to another space that at that same alpha value had the birth of 3 homology generators. But they would be sort of in some sense different homology generators, like they could be different non-bounding cycles, basically.
Mm-hmm.
And so the spaces could be really quite different things. So it's kind of a compression because you're just counting in some sense.
Yeah.
And is that impression correct? And more generally, this is a research question, is there any way that one could, instead of saying there are 3 that are born with alpha, when alpha is 0.6, could we say that there are 3 that are born, and this is kind of how large they are, or how big they are, or how many generators they have, or some other kind of characteristic that would discriminate more finely? And finally, is anyone else disturbed about this, or am I just being nitpicky?
I'm definitely also disturbed about this. I thought that was a very strange choice, actually, to just look at, you know, basically binning up and counting births and deaths, because like you said, it's not only a compression, but it's a compression of a compression, right? Like first you do topological invariance, and then you look at the barcode, and then you look at the binned features of that. So yeah, you kind of lose like a lot of information about which cycles are related to which boundaries. You basically lose the maps, and you're left with just some raw summaries of ranks of groups. So I think you could certainly capture more information if you went higher up. And like you could imagine maybe training, I'm not sure actually, maybe they tried this and it didn't work for some reason, but right, like why not learn on the barcodes themselves instead of binning and then tiling into some 2D thing? Like the barcodes already are kind of easy enough to convert into an image-like input. So that would seem like a natural thing to try.
Yeah, for us is a good comment there. So they're, you know, it's a strange combination in a way because they have all this math, but they're also trying to use the hammers that are available in their laboratories. They want to use that. So it's a good point, for sure.
I think they could put the CNN, even if they wanted to use the CNNs, though, they could put that on the barcodes instead of on the bin features.
Do image analysis on this barcode. It's a very reasonable thing to think about, for sure.
Yeah.
I don't understand why they couldn't use, like, 'cause for the barcodes, couldn't you just use like the starting point, like some starting point and then the length, and that would be sufficient? Like that sufficiently represents the barcode, right?
Yeah, so I absolutely, you could have different features. I mean, it'd be equivalent in some sense. But what I was worried about is the lack of sort of correspondence. So I guess one way of thinking about it is that, well, here's an example. I mean, probably a topologist could criticize this one. So suppose we had two different spaces, and one is a torus, so it's the surface of a bagel, right? It's got a hole in the middle and so forth. So, and the other space that I have is two spheres, just the surfaces joined together at a single point. And these are kind of different spaces in some sense. And I can look at various homology generators in there. So one on the tire or on the bagel or the donut, I can have one that goes around, around the small part and one part that goes around the big part. So I have two different circles, so to speak. And on the spheres, I can have one that goes around the left sphere, one that goes in the right sphere. And basically, now I do my persistent homology filtration thing, and I see that these generators are born and they die at a certain point. And you can imagine trying to form some kind of similarity or correspondence because each of these spaces has these two generators, at least for H1. I realize the spaces are different. But in some sense, in those spaces, there's very little similarity geometrically or topologically between those particular non-bounding cycles, right? So if you can see my hands, I have the bagel like this, right? It's a bagel to the surface of the bagel, and I have one cycle that goes around it like this, and the other cycle goes around it like this. And for the two spheres that are glued together at a single point, I have an equator here and an equator there. And I wouldn't want to make any generalization on the similarities of those spaces really based on the fact that perhaps the generators were born at the same time. But I think this method could. Or if it can't, it seems kind of anecdotal that they would actually turn out to be different based on these births and deaths. So hopefully I've just given an example that would give some intuition behind Ricky's very good comments. Any discussion?
Yeah, I've been thinking about this. So there's some really interesting algebraic topology in their work, and we've seen it before, like the 2017 paper. It seems like a very lossy function for what they're doing when they go into the machine learning phase of it. And the question I had was more of the form, is there actual biological intuition that we think we could glean from the Betty numbers and, well, even the birth and death counts if we could get that? Like, what are they measuring and how do we think it's important?
They talk about that a little bit in the paper. It's a bit, it's a bit hand wavy, the things that they present. But essentially they talk about hydrophobic interactions and this idea that the presence of a lot, what do they say exactly? They talk about how the presence of more holes and voids might indicate a less compact arrangement, or at least shorter-lived holes and voids might indicate a more compact, like hydrophobic network.
Couldn't they measure that more directly instead of counting birth and death counts?
So I'm referring to, this was all based on the barcode stuff, as far as I could tell. I don't think there's I don't remember reading any biological justification for why they featureize into the bin counts. I will mention that in the 2018 paper, mostly they use, I think they mostly use the barcode statistics as their 1D features. Those seem to perform better. So that I think corroborates some of the discussion we're having about this counts and bins being sort of a
Well, okay, so
question was
yeah,
justified.
yeah. The question I'm curious about, because I don't know the answer, is they do a lot of additional processing from structural data, and then they apply a machine learning neural network. What are they doing that's improving over the existing neural network infrastructure?
Maybe I don't. Can you expand more on that question? I think I don't
So
understand.
the straw man here is, why didn't they just throw structures into a neural network that was really big and see what happened? How is this better?
Yeah, I think their argument is something like they are. I mean, this is me completely putting words in their mouth.
Mhm.
But what I think their argument might be would be something like they are they're extracting, they're reducing the dimension of this geometric data, and they're doing it in a way that preserves some fundamental information about the space that they're in. And I think their argument is that those reduced features contain some biological information, perhaps related to
Yeah.
like interatomic forces and things like that.
So that's my understanding. And it raises the inherent conflict I see in this paper, which is, on the one hand, they say we're going to be very clever and devise a feature that will extract more information than just raw data. And then they apply the most brute force net method for raw data that they have to it in hopes that it'll extract information out of it.
Yeah.
I think this is a great discussion, and I think that very nice discussion and presentation. I'd like to sort of summarize two things that are puzzling to me about the paper. Two more things. So the first is, it's unclear to me whether the input for the testing are bound protein ligand complexes that have just been taken apart, or whether you're starting with the apo structures. And I don't mean to pester you with this, Graham, because I know you've been busy and flying across country and recovering from being sick and having house problems, but I'm really super curious about this answer, because I think if it's true that you can do this from two unbound structures, in other words, so again, this is the same problem we talked about before, but suppose I have a known protein complex A and B and I solve the co-crystal structure at high accuracy, and then I just take them apart and feed them into a program and ask how does A bind to B, and then I do that with a bunch of decoys. That's a substantially easier problem than if I've solved the apo, i.e. unbound crystal structures for A and for B in all their flexible glory, and then ask which proteins will mate. So I'd like to know that question. But if it's true that they can have this operate on the apo structures, then what happened to entropy, kinetics, and thermodynamics? I mean, that's telling us that essentially geometric, and I think topological with a star because it's kind of wind assisted, essentially geometric features are driving the ability to predict KDs, binding affinities, perhaps better than other methods without any explicit appeal to entropy or flexibility. And surely you couldn't model something, I think, that would require a big conformational change. But what I'm wondering is, how can they get that kind of accuracy without an explicit consideration of entropy?
Yeah.
So hypothesis one is that they're only doing it on the bound complexes, taking it apart, and they're preformed for binding, artificially, and so that you could just, it's a shape matching problem, which would make sense. But if they can really do it on the apo complexes, there's something very interesting happening. And I agree with both Ricky and with JJ, it may not be topology per se, but something interesting is happening because they're able to ignore entropy and get apparently good results. And both of these questions are mysterious to me. Any thoughts?
Yeah. Yeah, I agree. I think I wasn't able to, I mean, granted, I haven't read these papers with that specific question in mind, although it's a good question. I just checked the PDB-bind database. It looks like in principle, they have both protein ligand bound as well as protein apo structures. So I think in principle, it's definitely possible that they're using the bound structures. It's also possible that they use the APO structures. I didn't see any reference to that in the text, although, as I said, I haven't been specifically looking for that. Yeah, I think that's a really good question also. I know that they've done, I think they have another paper that they referenced, although I don't have it on hand. They applied these methods to try to look at frames of an MD simulation, and that, you know, I put that on my to read list because that sounds interesting, looking at how things change with conformational flexibility. But yeah, I think those are great questions, and unfortunately, I don't have the answers on hand. But
we could circle back. I think we have to move on in a minute, but I want to ask, does anyone else have any further comments or questions before we do? I want to take just a minute for a didactic point that may be useful. Graham, could you put up the slide of the sort of balls when you're defining the alpha complex again?
Sure.
And you just described this very well, but I want to just make a point that people might be able to take away. I thought your presentation was excellent. Great job. Please send us the slides. Very difficult material and nicely done. Yes, the one with that one there. Yep, just set that up there. So I want to just mention this because this work was developed at Duke by Herbert Elsbrunner and also Parkash to some extent. And so it's kind of like a local boy makes good. And I also just wanted to describe it using slightly orthogonal words. I think my words are saying the same thing as Graham, but you may remember this if you ever, because persistent homology and topological data analysis are very useful concepts. Already, I think 3 people in the class have used them in other classes at work, and that's an exponential increase from 10 years ago when this stuff was just coming online. So here's a very naive description of what this is really trying to get at, is that when you have this really complicated geometric object, like the one here, where you have a bunch of points and you have these balls and you have this graph here and so forth, just concentrate on this, on any of these edges, the straight things, or concentrate on the, just the straight lines of the graph. The idea is that when these are in space, they actually are located. It's not an abstract graph, they're embedded. So there's actually distances and stuff between these. And of course, that's in the paper. So we can compute topological invariance of these graphs, of this graph here, for example. We can count boundaries and cycles and things like that. But the point is that's a purely topological invariant. It doesn't rely on the geometry, and you want something that somehow links the topology and geometry. So sort of the genius idea of persistent homology, or these alpha shapes, is as follows. So just look at the graph here. That is the thing with straight lines. Now, imagine that I'm going to take a pencil that has an eraser of a fixed width alpha, and I can erase edges. Now, I can only erase, and I can also erase faces. Now, I can only erase it when my radius is small enough to fit between the 2 vertices, or for a face between the edges of the triangle. So I start out with my pencil having an infinite radius. So it can't fit between anything. So when I have the pencil eraser with infinite radius, I cannot erase any edges because it can't get between anything. But gradually, as I make the radius smaller, going down from infinity to 0, it can fit between 2 vertices. So for example, if 2 vertices are distance d, and my eraser is at distance r, which is less than that, I can get between them. So as I can reduce this radius alpha of the pencil, I can erase more and more edges and also faces and things like that. And basically the idea is that I want to graph changes in these topological invariants as I crank down this alpha. So when alpha is 0, I'm just left with all the points that are disconnected. And when alpha is infinity, then I have the original complex with nothing changed. And in the middle, I'm basically erasing stuff. And the idea is that you can't actually sample that or actually go from infinity down to 1, but using these algorithms, you can compute the critical values of alpha, namely the critical values at which something changes. And that's sort of the interesting thing in how they kind of try to wed geometry and topology. And Graham, you said something very similar, but I think the eraser model is one you can take away.
Yeah, I really like that explanation.
And if that didn't make sense to anyone, I'm happy to kind of talk about it. I think there's a little more in ASMB book, chapter 50. With that having been said, are there any more questions for Graham or anyone on this topic?